Welcome to this YouTube channel. Today we are going to discuss one of the forces of evolution, that is genetic drift. Friends, this video lesson consisting of the forces of evolution developed in three parts. First part already dealt with reference to the mutation and gene flow. This is the second part dealing with the genetic drift, and the next video lesson will come on the natural selection. Let us get started. The part 2. Contents. In the part 1 we discussed mutations and gene flow. This is the part 2. We are going to discuss genetic drift with subheadings founder effect and bottleneck effect. The next video lesson, part 3, will come explaining natural selection with subheadings stabilizing selection, directional selection, and disruptive selection. Modern synthetic theory or genetic theory of organic evolution, otherwise called neo-Darwinism, explains that small genetic changes that occur because of mutations gradually accumulate because of natural selection, and as a result of that, a process of evolution goes on. Not only the natural selection that is completely responsible for the evolution, there are certain other forces like mutation, gene flow, and the next one, that is genetic drift. These are also playing a very important role in the process of evolution. The only difference what we notice between the natural selection and other are, in natural selection, a positive variation with a good adaptive value is selected. Whereas in all other cases, randomly a change happens, and if that change is fit, then it will be selected. So that is the main difference. However, to understand the process of evolution, we need to understand Weismann's germplasm theory, which condemns the inheritance of somatic variations. Inheritance of somatic variations do not occur, but whereas the germplasm variations are only inherited. Hugo de Vries explained about mutations. Mutations are replacing the variations of Darwin theory, and Mendel laws of inheritance are substituting a Darwin law of pangenesis theory. So all together, these ideas, along with the Sewall-Wright genetic drift idea and other ideas, mingled here to construct or synthesize a new theory of evolution that is known as modern synthetic theory. As it is explained in terms of mathematics and genetics, this is also called genetic theory of evolution, and this theory is supporting Darwinism. Hence, this theory comes under neo-Darwinism. Friends, several scientists contributed to synthesize this theory. Modern synthesis, the phrase is coined by Huxley. Ronald Fisher developed several mathematical models to understand genetic theory of natural selection. J.B.S. Haldane introduced the concept of cost of natural selection. Sewall-Wright elucidated the natural selection and adaptation in genetics and the origin of species. The very important thing what we have to notice is the comment made by Dubzhansky. Say, as his comment goes on like this: mutations once seen as a rival to selection, but later it is the actual cause that is supplying raw material for the natural selection by creating diversity. So, what does this mean is, Charles Darwin simply discarded sports of nature. Sports of nature, otherwise called saltations, Charles Darwin discarded them. Charles Darwin felt that they are not very important for evolution process. and he concentrated only on small somatic variations. So, a big criticism on Charles Darwin was he explained survival of the fittest but not arrival of the fittest. But how the small variations actually arise? It was later answered by mutations. Once the mutations were thought to be acting as a rival to selection, but actually they are favoring the process of evolution by providing genetic diversity in the form of mutations. Okay, friends, Malthusian competition, mutations, Mendelian inheritance, and with all other ideas, we are now understanding modern synthetic theory. Modern synthetic theory also addressing the relationship between broad changes that is called macroevolution as observed by certain paleontologists, and also small minor changes that are happening in living local populations. It will address the two things, large variations or broad changes and small changes. The process of evolution is being driven by certain forces. These forces are driving the process of evolution. One species is driven to become other species because of certain forces. The forces are mutation, gene flow, genetic drift, and natural selection. But before these to be studied, we need to understand what is the basic minimal thing that the organism is having to undergo evolution. These are called the basic factors. Every living species which is performing sexual reproduction is having these basic factors to eligible for undergoing evolution. The basic factors are gene mutation, chromosomal mutation, genetic recombinations, natural selection, and reproductive isolation. They are the main factors. Okay, they are responsible for undergoing evolution, but all these need a kind of force to be driven, and these forces are called forces of evolution. Okay, I repeat, the forces of evolution are mutation, gene flow, genetic drift, and natural selection. In this video lesson today, we are going to discuss the genetic drift. Genetic drift was explained by Sewall Wright. Hence, genetic drift is also called as Sewall Wright effect. What is Sewall Wright effect? A simple definition. Any deviation from Hardy-Weinberg equilibrium by chance is called genetic drift. What is Hardy-Weinberg equilibrium? Hardy and Weinberg explained the equilibrium of allelic frequencies. The allelic frequencies always remain in an equilibrium for a gene. The genotypic frequencies remain always in an equilibrium for a gene. As long as a population is exhibiting Hardy-Weinberg equilibrium, it implies that the population is not undergoing evolution. Or else, if Hardy-Weinberg equilibrium is disturbed, then we need to understand that the population is undergoing evolution. But how is this Hardy-Weinberg equilibrium got disturbed? One of the disturbance occurs by genetic drift. Genetic drift is a deviation in Hardy-Weinberg equilibrium by chance. This is very, very important. It's a random deviation. By chance. Okay? By chance, we have a situation so that what is that? The equilibrium gets disturbed. Okay. So, a change in gene pool of small populations strictly by chance is also called genetic drift. In a small population, you see, for a small population, there is a small gene pool. So, in this gene pool, if any disturbance occurs, the Hardy-Weinberg equilibrium gets disturbed. This is called a genetic drift. One point we need to understand that genetic drift occurs strictly strictly by chance. Strictly by chance. Okay, so hence it is also known as aimless evolution. Sometimes it is described as an aimless change because it occurs by chance, not by choice. Okay, a change in gene pool of a small population is strictly by chance is called genetic drift. Evolution is a change in allelic frequencies in a population. So the modern definition, the most advanced definition of evolution is that a change in the allelic frequencies in a population is called a small evolutionary event. Okay, what are the chances here that are playing a very important role in bringing genetic drift and causing disturbance to Hardy-Weinberg equilibrium? They are the natural disasters like floods, forest fire, volcanic eruptions, and tsunamis, and several other natural calamities. Okay, so chance alone may eliminate certain members because of natural disasters, because of floods, because of forest fire, or because of volcanic eruptions, or because of tsunamis. Some very important people which are having critical genes, such members may be eliminated by chance. When the people having very critical genes are eliminated by chance, the population loses such genes. When population loses such genes by chance, its allelic frequencies get disturbed. When allelic frequencies get disturbed, Hardy-Weinberg equilibrium also get disturbed. That lead to a kind of evolution, that lead to change in the gene pool. Okay, so what change that has happened because of genetic drift may not be useful. It may be useful or it may not be useful. So we cannot give any guarantee that after the genetic drift, if a new population is formed and the new population is having an adaptive value, it's only a change. It's only a change. Whether the change is useful or harmful, it doesn't matter, but change has occurred. That is called genetic drift. So genetic drift is simply occurring by chance, strictly by chance. Okay, so now let's see. A gene which is representing 100% of the gene pool in a population, if gets disturbed or disappears, if it disappears, a gene disappears by chance. You take, for example, the animal which is having this particular gene, a group of animals which are having this particular gene happen to a mishap or subjected to an accident, met with an accident. So all such people if die, then the gene is completely eliminated from the population. So as it gets disappeared, it will cause disturbance in Hardy-Weinberg equilibrium. So the original population and newly formed population is having a difference with reference to this particular gene. Disappearing that particular gene may be advantageous or may be disadvantageous. So that we cannot give any guarantee. Genetic drift is causing evolution, no doubt. But there is no guarantee that the new population will be more fit than the original population. Change happens, that's all. We need to understand like that. So if you can understand natural selection as explained by Darwin, after natural selection only the fit will come out. The fit value, the variation which is having more adaptive value will be selected. But here anything that is changed is selected. Okay, there is no guarantee that the new population will be more fit than the original one. Okay, hence genetic drift is often described as aimless evolution. Sometimes it may be useful or it may be harmful. Okay, and genetic drift is having no adaptive value. It is non-adaptive evolution. It is aimless evolution. It is the evolution that occurs simply by chance, not by choice. Okay. So Darwinian natural selection brings a change by choice, but genetic drift brings a change by chance. That's the thing we need to understand. Genetic drift causes gene pools of two isolated populations to become dissimilar. Okay, two isolated populations, because of genetic drift, some people may die. So as a result of that, the two populations may become dissimilar. Sometimes because of a gene flow. Okay, because of a gene flow, all of a sudden, by chance, one group, one population, the donor population, may lose a particular gene. This is called a loss by chance. And the individual joins the new population. As a result of that, a particular gene is added to this population. This is called fixation by chance. Anything that happens strictly by chance is called genetic drift, according to Sewall-Wright. Hence, this is also described as Sewall-Wright effect. Okay, so genetic drift occurs in two methods. One is the founder effect, and the other one is bottleneck effect. First, let us discuss the founder effect. Founder effect is a case of genetic drift in which rare alleles or rare combinations of alleles occur in higher frequencies in an isolated populations. Say there is an original population, and some members from these original populations migrated to a new area and established their own population. When the process continues for several years, we will see the two populations. One is the original population and the other one is the new population. We expect that the new population is resembling original population, but in reality it is not. The new population, no doubt it is formed from the maternal population, mother population, but the new population does not resemble its original population because the new population is established by a few individuals of original population called founders or colonizers. Because of these few individuals only, a new population developed. So the new population will have the characteristics of founders, but not all representing individuals of original population. Okay, a new population developed from a few members of the original population only. So it will represent only the few members gene pool, not the entire mother population gene pool. So that's why a new population is formed because of founders. Founders are also called colonizers. Okay, when a new population is established by few individuals, who are called founders or colonizers of original population, such a new population slightly differs from its original population because founders carry a certain fraction of the total genetic diversity of original population. The original population total genetic diversity is not there with these founders. They are having only a part of genetic diversity. Because they are moving, only that part will move, not the entire part of the original population. So this is called founder effect. The alleles carried by founders are determined by chance. Only these members by chance went to that particular area to establish a population. Okay, if not these founders, if other founders would have gone, the population would have developed in new characters. So what the founders are moving, based on those founders only, the new population will get the characters. So the new population will not totally resemble the original population. You take, for example, many species on islands. When islands are discontinued by water, and when the terrestrial animals are living on islands, because they are discontinued, so only certain individuals from one island go to the other island to establish a population, a new population. that new population definitely resembles the founders only, but not the entire population. Those founders which are moving will have certain portion of the original gene pool, the entire gene pool. So as a result of that only, that population will be established. You will see good examples in Galapagos Islands, as Darwin observed these things. Galapagos Islands or many islands which are discontinued by water exhibit diversity. Exhibit diversity of different organisms like finches, tortoises. So, okay, the finches, what are present in different islands, are having different shapes of their beaks. The shape of the beak of a population of finch living in one island totally differs from that of other island. And the beak shape changed because their foraging techniques are changed. Their feeding habits are changed. To avoid competition, to avoid food competition for food, they changed their food actually. So when they are seed eating, actually, when they went to other area, more competition is there for seed eating. They changed their habit of eating worms. In that way, there a modification occurred either by chance or by anything. Okay, so these kind of changes of finches or these kind of changes in tortoises are correlated with their vegetation. That means food and environmental conditions in those islands. Okay, friends, what are those organisms that went on to new island to establish the population? New population resembles only those individuals, but not the entire individuals of original population. Okay, several human examples are also there to illustrate founder effect. The Amish population in US, United States, there is a particular group of population that is called Amish population, characterized by presence of polydactylity. The polydactyly, we have 5 fingers only, but these people are having more than 5 fingers, more than 5 toes. It is called Ellis Van Creveld syndrome. Okay. Why Amish population, individuals in Amish population are exhibiting polydactyly? It is because this population is established by certain founders who were having polydactyly, but in their original population, all the individuals were not having polydactyly. Only a particular portion of individuals are having polydactyly. Those individuals happen to migrate to a new area to establish the population. That's why the new population is resembling polydactyly founders, but not the pentadactyl individuals. So this is the best example to understand the founder effect. Okay, so this trait of having more digits is common in Amish communities in United States than in the entire United States population at large. The entire United States population is not having polydactyly. Only a sect of population is having polydactyly. Those people became founders for Amish populations. That's why Amish population is resembling these founders with the polydactyly. The other example you see in South Africa, in South Africa, a group of certain locality, Afrikaner populations exhibit a very, very rare phenomenon of Huntington's disease, because the founders just happen to carry this gene to this particular area and establish the population. Because the founders had the gene for Huntington disease, all their descendants are now having a gene for Huntington's disease. This is the best example to understand founder effect. You take, for example, Sri Lankan population. Sri Lankan population is very, very close to Mother Indian population. It does not resemble, Sri Lankan population does not resemble the morphological characters of all Indian population, but resembles only the South Indian people, a particular area of South Indian people, because the Sri Lankan population might have established by the founders traveled from this area. Okay, Indian population is consisting of the Indian gene pool is consisting of all members of all states, but only a particular area people went to that particular island and established the population. So that population resembles only their founders, but not the entire individuals of the population. Okay. The other best example that you can see is Red Indian population. In America, Red Indian populations are having only O positive blood group, ORH positive blood group. Why do they have ORH positive blood group? It is because their founders had the same blood group. Their original population where from the Red Indians came are having all possible blood groups, but only founders with the O positive blood group entered to this particular area to establish the population. That's why this population is exhibiting O positive character. So this is the way in which we need to understand the founder effect. Founder effect is bringing some changes from one population is becoming a new population with certain change. So this change is happened by chance as the founders migrated to that particular area by chance. Okay, the second very important event, or the second very important thing that brings genetic drift is bottleneck effect. What is a bottleneck? You just observe a bottle. So the bottle is very broad at the base and gradually it becomes narrowed. When we go towards the neck, the bottle gradually becomes narrowed. Likewise, a population bottleneck, original population is having broad gene pool, but when that original population is gradually moving towards evolution, the population moves towards bottleneck, so that its gene pool gets reduced. How the gene pool gets narrowed? The gene pool gets narrowed because of certain natural calamities, because of certain natural disasters, because of predation, because of hunting, because of habitat reduction. Okay, because of all these very important natural disasters, that the persons in this very broad gene pooled population, important persons are subjected to death. Important persons are subjected to death. As a result of that, the gene pool got constricted. As the gene pool reduced, then it will become a new population. Okay, so when certain people killed because of natural disasters, or because of predation, or because of hunting, or because of habitat reduction, this is not only for human beings but also for all other animals, the gene pool of the original population gradually becomes less. Okay, as a result of that, important genes are lost. Okay, important or unimportant, we don't know. The adaptive genes may be lost. So as a result of that, the population becomes very weak and it will become a new population. Okay, in terms of evolution, the population bottlenecks cause severe reduction in the number of individuals in population that leads to reduction in genetic diversity of the original gene pool. Okay, the populations may recover after having becoming bottlenecks. Their population reduces by the killings of individuals, by the loss of individuals. A very large population will become a small population, and this small population happened to live in that particular area and increase the population. Though the population increased after bottleneck, only the number increases but not the diversity. only the number of the population increases. The population may recover to their typical size, but not the same allelic frequencies. Original population allelic frequencies and bottleneck population allelic frequencies are totally different because some individuals are lost by natural disasters. So that you will find a new population. As the change happened in allelic frequencies by chance, it affects the future evolution of the species. So loss of individuals in bottlenecks will affect the future evolution process. Bottlenecks are more likely in relative large animals and plants than in smaller ones. Why? Bottlenecks occur only in those populations with large-sized organisms, not in small-sized organisms. The reason is large-sized organisms are less in number actually. Less in number indicating small size of population. The smaller the size of the population, the more will be the effect of bottleneck. Okay, if 100 individuals are there, if one individual is lost by bottleneck, the effect is very less. If 10 individuals are there and one individual is lost, then it will have greater impact. That is why bottlenecks are more likely in relative large animals and plants than in smaller ones. Primitive human populations were in small groups and most of them are decimated by disease, killed by disease, by war, or killed by other natural disasters. When the primitive humans are in small populations and some individuals are eliminated, decimated by means of disease or by means of war or by means of natural disasters, this loss can affect the allelic frequencies. Okay, so we can observe today the blood groups like A, B, O, A group, B group, O group. So the evolution of such A, B, O blood groups might have arisen at least in part as a consequence of bottleneck in our ancestral populations. Say the best example to understand bottleneck in the present situation is to observe the cheetah population. The cheetah population has very less genetic variation in its gene pool. Cheetah is very, what is that? It is feeling very shy in the reproduction event. A female cheetah copulate with many males in order to make their progeny survived, in order to make their progeny not killed by a male member. Okay, so it is adapting a new situation, a new thing. But this cheetah population, the gene pool of this cheetah population is very less because this population of cheetah might have experienced a bottleneck around 10,000 years to 12,000 years ago during the Ice Age. During the last Ice Age, most of the individuals of cheetah population might have decimated, died. As a result of that, in today's cheetah population, the size of the gene pool is very small. In such a small gene pool sized population, if another bottleneck occurs, it will be a disastrous thing in evolution. The population will suddenly be changed. Okay, and another best example to understand bottleneck is elephant seals. So elephant seals of north side and elephant seals of southern side, northern elephant seals. Let us classify northern elephant seals and southern elephant seals. The northern elephant seals are subjected to hunting by people, so that they got reduced genetic variation due to population bottleneck as humans inflicted on them in 1890. During 1890, more hunting occurred on northern elephant seals. Hunting reduced their population size to only 20 individuals at the end of 19th century. Because of hunting, their number got drastically reduced. Northern elephant seal number got drastically reduced because of human activity, anthropogenic hunting. These 20 individuals what remained, now they increased the size of population and have become 30,000 individuals. The population now is very big, but population is big in the form of in the number of individuals, but not in the number of genes. Gene pool is very small, population is very large, and such a difficult situation is said to be a new population with new changes, and this happened because of bottleneck. But the same elephant seals which are living in the southern area, southern elephant seals, are not affected because they were not hunted by human beings. So, certain natural disasters like hunting, habitat reduction, and floods, and certain other things like volcanic eruptions, and it may in human beings it may be certain superstitions, and all these are causing bottlenecks. What is a bottleneck? A drastic reduction in the size of gene pool. A drastic reduction in the size of gene pool will cause a change in the population. A change in the population is the basis for the formation of evolution. So evolution occurs by the genetic drift by chance. Friends, you might have understood the genetic drift, which is having two parts. One is called the founder effect, where the founders are establishing a population, and the bottleneck, the natural disasters which remove certain very important individuals, thereby reducing the size of the gene pool. Hope you have understood this one, and in the next part of the video lesson we are going to discuss natural selection as a very important driving force of evolution. Thank you friends. Thank you for listening to this video lesson.